let's move this to our second to last, if you can believe it, second to last speaker of the day, Adam Kaplan, who is the Chief Scientific Officer at MYMD Pharmaceuticals, ticker there, MYMD. Oh, man, I see a big old black screen where Adam's face was just a second ago. I'm wondering if he lost the internet. You know what? I bet you that's what happened. Let's see if he's I've seen this before when this happens on the service that we use where when someone loses internet, it actually happened yesterday, and then their face is gone. And so I think that's what happened with Adam. Is he just lost his internet? So we'll get Adam back here in a moment. I promise. We are only 3 minutes behind schedule, which is a victory, right? It's a victory. All right. Let me do this for one of the last times, if not the last time today. Bring up the Slido poll, okay, that I've been mentioning throughout the day. There was the QR code. Scan that QR code. Answer the poll. You could win a lifetime Benzinga Pro subscription. Okay. Here's the poll question that I care the most about. But let's move off this into a different one. Let's move off this to here's a trivia question. Doge or Shiba? Doge or Shiba? I'm surprised. Oh, but then we got Adam back. So I'm surprised Doge is dominating this poll. I would not have thought that. But what do I know? Let's see if we can get Adam back here. Adam Kaplan is the Chief Scientific Officer. There he is of MYMD Pharmaceuticals, ticker MYMD, Mary, Yankee, Mary, Delta. Adam, we had you, we lost you, and now we have you again. So all is well.
Excellent. Thank you, Spencer, for bringing me back from the edge of oblivion there.
Let's go ahead, if we can, and share your presentation, and we can get right into it. Click on that share button. And yeah, I'm sorry? I do not see it. No.
Can you see the presentation?
I do not see it.
That's a problem.
You can click on that share button in the middle there. I had a feeling that you had some sort of internet outage. That's what typically happens. There, I see something now. There we go. We're in business.
Oh my goodness. That was quite upsetting. So, all right. Good afternoon, and thank you for joining this presentation to learn more about MYMD Pharmaceuticals and our two currently developing drugs, MYMD-1 and Supera-CBD. My name is Adam Kaplan, as Spencer has already alluded to. I'm the Chief Scientific Officer for MYMD. After leading Johns Hopkins last year, after being there for 25 years as a clinician researcher in areas related to autoimmune diseases. Before I begin, I'd like to call your attention to the customary safe harbor disclosures and forward-looking information, which I know, like me, you're looking forward to reading, or are you? The first thing you should know is that, as I will elaborate on shortly, MYMD-1 is an improved TNF-alpha inhibitor designed to be taken by mouth in comparison to currently available medicines, which are injectables and carry a black box warning for the risk of lethal infections. In addition, MYMD-1 has selectivity, and we believe will make it much safer to take, as I'll show you in just a bit. The TNF-alpha inhibitor global market size is $40 billion, with the lead compound Humira alone accounting for $20 billion of that market, which makes it the #1 selling drug in the world. So if you remember nothing else from my presentation today, please remember that what we have here is a potential blockbuster. The other take-home message I wanted to make sure you got from this presentation right up front is that we are on track to have a phase 2 study with results of MYMD-1 treating COVID-19 patients at Johns Hopkins by the end of this year, 2021. Now that hopefully I have your attention, I'd like to tell you more about MYMD-1 and the reasons we believe in its potential to impact a number of diseases through its unique mechanisms of action. MYMD-1 is an immune system regulator that has been designed to be taken by mouth and to regulate the release of inflammatory cytokines, including tumor necrosis factor alpha, which is abbreviated TNF-alpha. Cytokines are the chemical messengers that the cells of the immune system use to communicate and rally one another. They are like what neurotransmitters are to neurons, cytokines are to the immune system. TNF-alpha is the primary cytokine regulator responsible for inflammation in the body. TNF-alpha is one of the first responses of the immune system to an acute infection, but when chronically overactive perpetuates a cycle of immune dysregulation that can cause and perpetuate autoimmune diseases. Inflammation is either a beneficial consequence of the activation of the immune system to fight off foreign invaders or a telltale sign of an underlying illness that has taken hold. In addition to autoimmune diseases, diseases of old age are usually accompanied by an overactivation of the immune system and therefore accompanied by increased inflammation. Age-related diseases such as heart disease, lung disease, kidney disease, cancer, Alzheimer's, and any one of more than the 80 known autoimmune diseases all involve immune activation, overactivation, and dysregulation. Ultimately, what they all have in common is a cascade of mounting activation of cytokines resulting in inflammation burning out of control, and TNF-alpha is the spark and tinder that lights and perpetuates the fire. Before highlighting studies we have initiated to demonstrate the effectiveness of MYMD-1 to treat autoimmune and age-related diseases, let me describe for you now something about how MYMD-1 works. MYMD-1 works to target the cause, not the symptoms of diseases associated with immune activation and inflammation. At the doses we've investigated so far, there is minimal impact expected on cell viability, which translates into enhanced clinical safety. This is an important differentiating factor compared to the available TNF-alpha inhibitors which are on the market. MYMD-1 also crosses the blood-brain barrier, unlike available TNF-alpha inhibitors, which we believe will eliminate the known side effects of neurotoxicity associated with them. This slide demonstrates the way MYMD-1 inhibits multiple cytokines. So not just TNF-alpha, but you can see TNF-alpha, IL-6, and IL-17 are all inhibited by increasing doses of MYMD-1 in an assay developed and carried out by Eurofins on our drug. The two most common examples of overactivation of the immune system where TNF-alpha plays a key role are one, when the body starts to mistake itself for a foreign invader, leading to autoimmune diseases like multiple sclerosis, rheumatoid arthritis, and inflammatory bowel disease. And two, when the immune system gets completely overwhelmed and goes on a no-holds-bar assault, which is what researchers observe in the quote-unquote cytokine storm observed with COVID-19. The first-line response of the immune system to fight off invaders that have breached the walls of our skin and digestive tract is led by the immune system activation of TNF-alpha. While this function is essential when serving as the frontline coordinator of the immune system, TNF-alpha can wreak havoc when the immune system gets overheated, leading to effects gone out of control. As you can imagine, having a way of selectively preventing out of control injury, but preserving the ability to fight off infection would be optimal in a TNF-alpha inhibitor, and that is precisely what MYMD-1 is designed to do. Humira and the other currently marketed TNF-alpha inhibitors act by indiscriminately blocking TNF-alpha, including its role in coordinating the initial response to invaders, and can cause serious infections. In fact, the FDA currently requires a black box warning on all TNF-alpha inhibitors that warns of increased risk for developing serious infections that may lead to hospitalization and even death. And death is a side effect that you do not want on your package insert. MYMD-1, on the other hand, has been designed to be selective, to block TNF-alpha when it becomes overactivated in autoimmune diseases and cytokine storms, but does not block it from doing its normal job of being a first responder to any routine type of moderate infection. This means we believe it should be much safer. And hopefully you can see my cursor here. Black is the inhibition that happens here either with Humira or Remicade, TNF-alpha inhibitors. And here, this LPS is a model of bacterial infection in the system. And you can see that the TNF-alpha inhibitors block that, but MYMD-1 does not. However, the BT is a model of autoimmune diseases where lymphocytes have gotten out of control. And both the TNF-alpha inhibitors and in red, MYMD-1 are potent blockers of TNF-alpha. It is this selectivity that we think will allow the preservation of the ability of MYMD-1 to allow initial responses of the immune system, but only adjust what's going on with autoimmune diseases. In addition to its effect on the immune system, MYMD-1 has been shown the potential to create other unexpected but welcome effects on the body. MYMD-1 prevents fibrosis, which is the scarring that happens as the result of chronic injury from inflammation. Fibrosis is involved in injury to a number of organs, including lung, kidney, and liver, which together comprise an anti-fibrosis market size of $110 billion. In fact, it's even estimated that 45% of all deaths in the United States are involve fibrotic disease. So now let's transition to what we've already planned and started to move MYMD through to market approval by the FDA. We have both short-term and long-term plans for demonstrating the effectiveness of MYMD-1. Most autoimmune diseases are degenerative in nature, which means they progress slowly. Early and late-stage studies in multiple sclerosis, rheumatoid arthritis, and frailty, which is the consequence of poor physical adaptation to aging, all take 6 months to a year of treatment to resolve the difference between drug and placebo. This translates into a minimum of 9 months to 1.5 year long studies. These are long-term steps to develop novel therapies for chronic diseases, and the investment of time is necessary to achieve blockbuster breakthroughs. There are also short-term methods to demonstrate the potential impact of MYMD-1, and our company intends to exploit these opportunities. As it turns out, the brain is very sensitive to inflammation and acts as a canary in the coal mine. Either robust or chronic inflammation results in immune-mediated depression. Autoimmune diseases are associated, all of them, by the way, are associated with 25 to 50% rate of immune-mediated depression, which correlates with their level of TNF-alpha and IL-6. Blocking either one of these cytokines can lessen depression in autoimmune diseases, and MYMD-1 has been shown capable of blocking both simultaneously. Excuse me. Preclinical studies done just this week at Johns Hopkins demonstrated that MYMD-1 has the ability to treat immune-mediated depression in the lab, preclinically. I'm happy to talk about that in the question and answer if people are interested in how that was done. The short-term advantage of studying the effects of MYMD-1 on immune-mediated depression is that treatment for 1 month can be sufficient to demonstrate mood changes due to inflammation, making studies as brief as a few months sufficient to complete the study. Essentially, we are using immune-mediated depression as a readout to monitor the anti-inflammatory effects of MYMD-1, where the levels of TNF-alpha in the blood is still the primary outcome measure. COVID-19 is a global menacing once-in-a-century killer. And in the US alone, it's affected 33 million people and killed over 584,000 people. It is also a condition for which MYMD-1 might be ideally suited to assist with. COVID-19 exerts its greatest injury through causing a storm of cytokines release, leading in the worst case scenario to lung injury and even complete destruction. TNF-alpha correlates with the severity of the disease and lung injury, and the lung injury results in pulmonary fibrosis, both of which MYMD-1 has been shown capable of preventing in laboratory tests of human immune and lung cells. Not surprisingly, COVID-19 causes clinical depression in over 50% of those infected with SARS-CoV-2, which is the virus that causes the pandemic. Because both COVID-19 and the depression it causes happen quickly, 2 weeks of treatment are sufficient to test the ability of MYMD-1 to treat COVID-19, meaning that a study we have planned will take 3 months from the start to finish in order to complete. This study is under contract with Johns Hopkins, and we are expecting to have phase 2 data from COVID-19 study of using MYMD-1 by the end of this year. This is an example where you can see this is increasing doses of MYMD-1, and it potently decreased cytokine storm in the laboratory where you overactivate immune cells by both decreasing TNF-alpha, interferon gamma, IL-2, and IL-10. In addition to long-term study of MYMD-1 in progressive MS, for which there is a contract in place at Johns Hopkins, we are also planning a short-term study that will give us the more rapid readout of the effectiveness of our treatment. MYMD recently submitted an orphan drug designation to the FDA for immune-mediated depression and MS. Similar to our COVID-19 study, an MS-associated immune-mediated depression study affiliated with Johns Hopkins is planned to begin in the fourth quarter of this year. A long-term 2-year longevity study in mice with MYMD-1 versus rapamycin was conducted by the same scientist who published the MYMD-1 study on autoimmune thyroiditis from the Johns Hopkins. This is the study in the middle of the slide you can see here. The manuscript, which showed a 30% enhancement of lifespan by MYMD-1 that beat rapamycin, is expected to be submitted this summer, followed by publication in a peer-reviewed journal. This, combined with the known role of inflammation in contributing to the age-related decline, has prompted us to investigate the potential role of MYMD-1 in slowing aging. MYMD recently authored a white paper on aging and sarcopenia, which can be viewed from a link on our website. Sarcopenia is an age-related decrease in muscle mass, muscle strength, and muscle performance. Sarcopenia, in conjunction with cognitive changes, are part of the frailty syndrome, it's been called, that can occur with advancing age, which in turn implies additional risk in the morbidity and mortality of individuals. And this has been approved by the FDA as a target for aging-related research. There is no clear comparator for an indication in slowing aging and sarcopenia for MYMD-1 because no such drug with FDA approval currently exists. The market for MYMD-1, if it demonstrates the ability to substantially slow aging and/or muscle loss, is priceless. MYMD-1 has contracted with IQVIA, the global leader in clinical trial design and management, to assist with a phase 2 study of sarcopenia and frailty to be performed at Johns Hopkins beginning in the first quarter of 2022. Primary outcomes will include chronic inflammatory biomarkers, including TNF-alpha, that is inhibited by MYMD. Secondary outcomes will include gait and strength. All of these efforts will allow us to yield further evidence in our ongoing path forward to extend health span or a healthy lifespan. I'd like now to shift to our other candidate, which is equally exciting in its therapeutic potential that affects endocannabinoid system in a manner that is superior to plant-based cannabidiol, better known as CBD, about which people have heard a great deal today. Supera-CBD is a patent-protected synthetic CBD derivative that is being developed to address anxiety, chronic pain, and seizures. Supera-CBD is synthetic. It's a cannabidiol derivative that targets endogenous endocannabinoid receptor type 2, or cannabinoid receptor type 2, abbreviated CB2. Supera-CBD has broad therapeutic properties across a range of neuropsychiatric disorders, but without the intoxicating effect that is seen in THC. On April 27th of 2021, so just recently, new data from a study of Supera-CBD from the researchers at Johns Hopkins University School of Medicine was presented at the 3rd Annual Neuroimmunology Drug Development Summit. The preclinical study was able to highlight Supera-CBD's promising anti-anxiety properties, demonstrating that effect in mice, and a dramatically 4-fold higher binding affinity for the CB2 receptor that you can see here. And Supera-CBD was 92% compared to CBD 25% binding affinity. CB2 receptors are found in the immune system and have been implicated in a number of therapeutic effects of CBD, potentially including anxiolytic, anticonvulsant, antipsychotic, neuroprotective, and anti-inflammatory effects. Certainly the anti-inflammatory effects have a great deal of support for the role of CB2. Supera-CB2 is manufactured as a synthetically pure molecule, and as a new molecular entity carries with it patent protection for the next 2 decades. Plant-derived CBD has neither the purity, consistency, or exclusivity that comes with Supera-CBD. Demonstrated in this visualization of our current pipeline, MYMD is planning a phase 2 trial for sarcopenia and immune-mediated depression. Additionally, We are nearing the beginning planning stages of phase 2 studies for MYMD in COVID-19, multiple sclerosis, rheumatoid arthritis, and other autoimmune diseases. MYMD-1 is protected by a worldwide patent portfolio, which to date includes 11 granted patents and more than 20 pending applications. In closing, thank you all for your time and attention. I'm happy to field any questions that Spencer might have dug up for us. And we are extremely excited about the prospects of these two drugs and their potential therapeutic opportunities. So thanks for your time.
Thank you, Adam. And we are just about at time. Let me see. Let me go through a couple of our chats and let me move over here. I don't remember if you addressed this, so I apologize if you did. But can you elaborate maybe a little bit on the Humira comparison?
Yeah, absolutely. So thanks for that question. So Humira,
that's like the top. It's like the top selling drug in the world, right?
Yeah, that's correct. And in fact, even though it's no longer has the patent protection that it did, and it's been on the market, I believe, for something on the order of 15 years, it still is bringing in $20 billion. It doesn't have the exclusivity, but it has, the market share. And it is a TNF-alpha inhibitor, as is ours. However, ours, unlike all of the TNF-alpha inhibitors, ours is orally available. Ours crosses the blood-brain barrier, and all of these TNF-alpha inhibitors that do not cross the blood-brain barrier, all the available ones, most of which are antibodies or ways of sucking up and binding non-specifically or non-selectively all the TNF-alpha that they find, they all have the risk of actually causing neurotoxicity, such as this strange syndrome where patients on TNF-alpha inhibitors for rheumatoid arthritis, for instance, or inflammatory bowel disease, will get an MS-like condition for which we don't currently know why that is. But we know that if you can cross the blood-brain barrier, you should be able to prevent that from happening, and MYMD-1 does. And then finally, the selectivity that MYMD-1 has, where it doesn't affect what's called innate immune response. It doesn't affect, if you get infected with a bacteria, it doesn't block that TNF-alpha, but it does block the TNF-alpha that gets overactivated when you have autoimmune diseases and lymphocyte overactivation.
All right. Adam Kaplan, Chief Scientific Officer of MYMD, ticker MYMD on the NASDAQ. Adam, thanks so much. Learned a lot.